Good morning, everyone. Today, we'll be diving into the fascinating world of amino acids as neurotransmitters. I'm MS Nadeem, and I'll be guiding you through this presentation. We'll explore how these fundamental building blocks play crucial roles in neuronal communication. Let's start with the basics. Neurotransmitters are chemical messengers released by nerve cells. They transmit nerve impulses between neurons and to target tissues like muscles, organs, or glands. Think of them as the language neurons use to communicate. This slide illustrates how neurotransmitters are produced and stored. They're synthesized in the neuron's cell body, transported to the axon terminal, and then carefully packaged into synaptic vesicles, ready for release when a nerve impulse arrives. Here, we see the process of neurotransmission in action. A nerve impulse triggers the release of neurotransmitters into the synaptic gap. These neurotransmitters then bind to receptors on the postsynaptic neuron, initiating a new nerve impulse. Neurotransmitters can be broadly classified into three types based on their effects: excitatory, inhibitory, or both. Glutamate and aspartate are key excitatory neurotransmitters, while GABA and glycine are major inhibitory ones. Acetylcholine and norepinephrine can be either depending on the receptor. This table summarizes the effects of key neurotransmitters. For example, glutamate excites neurons and is involved in memory. Imbalances can lead to migraines or learning issues. GABA inhibits neurons. Too little can cause seizures, while too much can cause anxiety. Initially, amino acids weren't considered prime neurotransmitter candidates. This was because they're common components of all cells. However, we now know that they play a vital role in generating excitatory and inhibitory synaptic potentials in the CNS. Here's a visual representation of amino acid neurotransmitters. Aspartate and glutamate are the primary excitatory neurotransmitters, while glycine and GABA are the main inhibitory ones. Notice their distinct chemical structures. With the exception of GABA, most amino acid neurotransmitters are intermediates of protein metabolism. GABA, however, is unique. It's synthesized by glutamic acid decarboxylase, GAD, an enzyme found almost exclusively in neurons. Glutamate biosynthesis occurs through two main pathways. One involves the reductive amination of a ketoglutarate, and the other involves aminotransferase reactions. This diagram highlights the first pathway, showing the conversion within the TCA cycle. This slide details glutamate biosynthesis in the presynaptic terminal. Glutamine is converted to glutamate by glutaminase. The blood-brain barrier is impermeable to glutamate, so it must be synthesized de novo from glucose. Glutamate transporters, or E8s, are crucial for regulating glutamate levels in the synapse. There are 5 types, each with a specific location. For example, EAAT1 and EAAT2 are primarily found in astrocytes. Glutamate receptors are diverse and can be broadly classified into ionotropic and metabotropic types. Ionotropic receptors include AMPA, NMDA, and kainate receptors, while metabotropic receptors are divided into 3 groups. Ionotropic glutamate receptors are ligand-gated ion channels. When glutamate binds, the channel opens, allowing ions like Na+, K+, and K2+ to flow through. This generates an excitatory postsynaptic current, triggering an action potential. This diagram illustrates the relationship between glutamate and its ionotropic receptors. Glutamate is the neurotransmitter, and AMPA, NMDA, and kainate are the agonists that activate their respective receptors. Let's delve deeper into the types of ionotropic glutamate receptors. NMDA receptors are selectively blocked by APV, while AMPA and kainate receptors are blocked by CNQX. This distinction is important for pharmacological studies. NMDA receptors are unique because they are both voltage-gated and ligand-gated. This diagram shows the receptor structure, including the GLUN1 and GLUN2 subunits and the binding sites for various molecules. The glutamate-glutamine cycle is essential for maintaining glutamate homeostasis. Glutamate released into the synapse is taken up by glial cells, converted to glutamine, and then transported back to the presynaptic neuron. This slide illustrates the function of the NMDA receptor. The influx of Ca2+ through the open channel activates calcium-dependent signaling cascades. The channel is blocked by Mg2+ in a voltage-dependent manner. AMPA receptors are found in all neurons and are composed of 4 subunits. Most contain GLUA2 subunits and are permeable to Na+ and K+, but not Se2+. Aberrant trafficking of AMPA receptors is implicated in neurodegenerative diseases. AMPA receptors are highly mobile proteins that undergo translocations. They require a high concentration of glutamate for activation compared to NMDA receptors. They also have multiple binding sites. Here's a comparison of AMPA and NMDA receptors. AMPA receptors may or may not be calcium permeable and are not blocked by magnesium ions, while NMDA receptors are calcium permeable and blocked by magnesium ions. Agonists activate receptors, while antagonists block them. This slide lists NMDA antagonists like AP5 and CPP, and AMPA antagonists like quinazoline 4-1 and perampanel. These are valuable tools for studying receptor function. Kainate receptors are another class of ionotropic glutamate receptors. They modulate neurotransmitter release, depolarize the postsynaptic membrane, and regulate neuronal excitability. Unlike AMPA or NMDA receptors, KRs are widespread in both post- and presynaptic regions. Presynaptic kainate receptors play an important role in modulating neurotransmitter release. Dysfunction of KRs has been linked to several diseases, including temporal lobe epilepsies, schizophrenia, Huntington's disease, and Alzheimer's disease. KRs antagonists are used for the treatment of epilepsy. GABA receptors are found throughout the brain and spinal cord. GABA is the major inhibitory neurotransmitter in the mammalian brain, playing a crucial role in regulating neuronal excitability. GABA is synthesized from glutamate by the enzyme glutamic acid decarboxylase, GAD. This reaction also produces CO2. This is a critical step in the production of this important inhibitory neurotransmitter. GABA acts as an inhibitory neurotransmitter in the brain. There are 2 main types of GABAergic receptors: GABA-A, which are ligand-gated chloride ion channels, and GABA-B, which are GPCRs. GABA-A receptors have a pentameric structure composed of 5 subunits. This diagram shows the receptor embedded in the cell membrane with binding sites for various molecules like GABA, benzodiazepines, and barbiturates. This slide further illustrates the pentameric structure of GABA-A receptors, highlighting the different types of subunits and their functions, such as involvement in sleep, anxiety, and muscle relaxation. Benzodiazepines enhance the effect of GABA by increasing the frequency of chloride channel opening. This leads to hyperpolarization of the neuron, reducing its excitability. The pharmacological actions of GABA-A receptors include anxiolysis, sedation, amnesia, anticonvulsant effects, and muscle relaxation. These effects are mediated by the influx of chloride ions into the neuron. This slide lists different types of drugs and their effects on the GABA-A receptor. Barbiturates, benzodiazepines, and Z-hypnotics facilitate GABA-A action, while flumazenil is an antagonist at the BZD site. GABA-B receptors are metabotropic and coupled to GI proteins. This leads to a decrease in CAMP, inhibition of Ka2+ channels, and activation of K+ channels. GABA B receptors inhibit neuronal activity by decreasing Ca2+ influx in presynaptic neurons and increasing K+ outflux in postsynaptic neurons. This leads to hyperpolarization and reduced neurotransmitter release. Baclofen is a selective GABA B agonist and muscle relaxant. It is used to treat spasticity and skeletal muscle rigidity. However, it can also cause side effects like sedation and weakness. Thank you.